Good morning. Study session one of module three. But in brief, the content of module three includes sources of amino acids, which we are going to be discussing today. Study session two talks about the disorders of amino acid metabolism, some disorders really. Study session three talks about diabetes mellitus. Of course, you are nursing students, so you should know about this, right? Diabetes. You treat diabetic patients all the time, don't you? All right. Study session four, physiological chemistry. Study session five, pathological chemistry. Okay. All right. Starting with study session one, sources of amino acids, fits. We're going to look at sources of amino acid, fits of free amino acids. All right. Digestion of dietary protein, degradation of endogenous proteins, catabolism. That's the breakdown of amino acids. Okay. All right, this is just a basic introduction. What are amino acids? They are monomeric units of proteins. Amino acids are what make up proteins. Just like polysaccharides are made up of monosaccharides, simple sugars, glucose, mannose, galactose. They make up your starch, your glycogen, your pectin, your cellulose, your hemicellulose. You understand? So you get polymers from monomers. Protein is a polymer, all right? Amino acid is the monomer that makes up the polymer protein. So I hope you understand this simple explanation. Metabolism of amino acids involves the breakdown of amino acids and how amino acids are synthesized. Basically, that's what metabolism is: catabolism and anabolism. In catabolism, it will be breakdown of something, either breakdown of proteins to amino acids, or anabolism will be synthesis of proteins from amino acids, right? I think it makes sense. Okay. All right. Basically, this is just a short introduction. Now, sources of amino acids, right? What are sources of amino acids? We obtain amino acids from dietary proteins, mostly from food, right? We also derive amino acids from endogenous breakdown of enzymes, receptors, and other cellular proteins. Doesn't this make sense? You can get amino acids from food or from breakdown of proteins, cellular proteins, proteins in the cell in your body, or receptors. Some of these receptors are proteins, glycoproteins. They contain proteins that could be broken down into amino acids. You know what receptors do, right? You should. Enzymes. Most enzymes are proteins, but not all proteins are enzymes. Do you understand? There are enzymes that are not proteins, but most enzymes are proteins. Okay? So most enzymes are proteins except what we call ribozymes. Ribozymes are RNA molecules capable of catalyzing reactions, but they are not proteins. But most enzymes are proteins. So you can get amino acids from breakdown of proteins in the body. Okay? All right. So the third sources of amino acids can be through direct synthesis. That will be the body synthesizing its own protein, or chemical synthesis in the lab. You understand? So this is limited to essential amino acids only. The body could synthesize. No, I think this statement here is wrong, because the following statements will prove the error in the initial statement. You see, your diet contains proteins and amino acids, okay? The amino acids that come from your diet are non-essential since you can get them. But amino acids, sorry for this mistake, please, okay? Amino acids that you get from diets or you get from an exogenous source, those are essential amino acids, okay? Okay, okay, because amino acids that can be synthesized by your body are not essential. Of course, they are needed, they are much needed, but since they can be easily synthesized by your body, you can't call them essential. Those that your body cannot synthesize and you need to get them elsewhere, those are essential amino acids. I think this makes more sense, okay? All right, so now let's show this, so that you get an idea of what we're talking about. I've I think, shown this slide multiple times. We come to where you discuss your proteins: milk, yogurt, cheese, right? Milk, those dairy products, they're also rich protein sources, right? Meat, poultry, fish, egg, beans, and nuts. These are also rich protein sources, right? So, so it means you can get amino acids from this food. As they go in, there's a breakdown, catabolism of proteins to yield necessary amino acids. Basically, that's what we are seeing. All right. Now, what are the fates of free amino acid in the body? After your body is done utilizing the amino acid it needs for a particular metabolic process, excess amino acids, what do your body do to them? Okay. So the primary source of free amino acid in the body is the breakdown of dietary protein, right? Now, free amino acids are used mainly to synthesize proteins, which I've said this before, that will be anabolism, right? Okay. But they can also be used as precursors for a variety of nitrogenous compounds. Of course, why? Because look at the basic structure of an amino acid. This is it. The amino acid has the alpha carbon, which is the center. This is the chiral carbon in chemistry. All right. It has a carboxyl group. It has an amino group. Okay. So this amino group can help. This amino group is what makes an amino acid an excellent choice for like for use as a scaffold for a variety or precursor for a variety of nitrogenous compounds. Okay. All right. So, and this is the R group. Now, most amino acids differ in this R group. Okay. So the R group will contain something else that will make one amino acid different from another. For example, alanine will have a CH3 here. Valine will have more aliphatic group, not just CH3, for example. I'm going to show you a chart showing the structures of amino acid. You're going to get what I'm saying there, okay? So they also serve as precursors for a variety of nitrogenous bases, and we know why, because of this amino group that's present in the basic structure of an amino acid, okay? Amino acid present in amount exceeding the quantities needed by your body when you have excess and cannot be stored by the body. They can be degraded to yield ammonia, okay, which is going to be converted to probably urea and excreted from the body, or intermediates of carbohydrate and lipid metabolism. I think we remember very clearly when we discussed glycolysis. You look at this chart I showed you back then. Let me show you again. Okay, not this. Right, this is the chart. Yes. Okay, you remember amino acids, you have them in excess. Some are probably converted to ammonia, which will enter the urea cycle and be excreted from the body. Others could be converted into precursors of a carbohydrate metabolism. For example, glutamine can be converted back to alpha ketoglutarate, which is an intermediate in the citric acid cycle, okay, which can be converted into energy or converted through the glycosylate cycle and gluconeogenesis into glucose, okay? Oxaloacetate, aspartate, I think asparagine aspartate could be converted to oxaloacetate, if I'm not mistaken. Please, you can confirm that. All right. So some of these amino acids can serve as precursors, in some of these metabolic reactions. All right. Okay. All right. Yes, exactly. This was what I just finished saying. Okay. You see, proline, glutamine, glutamate can be converted to alpha ketoglutarate. This is an excellent chart. Aspartate, asparagine can be converted to oxaloacetate. So this makes sense, doesn't it? So there's no waste in the cell. There's a cycle. The body operates on the principle of maximum economy. Okay. So economy is what happens in a sustainable system. Even in our country here in Nigeria, if we try to promote sustainability and economy, you think things would be better. But extravagance, theft, greed will not allow us to do so, right? Okay. So basically, this is what we are saying. The intermediates can be converted to glucose, which I've explained already, or fatty acids, or oxidized to yield energy. Of course, ammonia is used to synthesize glutamine, of course, the principal feed of free ammonia and glutamate. Free excess ammonia is used to make urea, which is excreted in the urine. I think this is quite self-explanatory. Now we have to talk about dietary protein. How does the body break down proteins for break down proteins and produce amino acids, which is needed by your body? I think this is probably something you know, really, but I'll just try to go at it briefly. You have to take your time at it. Usually when you take in proteins, okay, the digestion of proteins begins in the stomach, okay? The gastric mucosa contains hydrochloric acid. Now, the protein in the stomach that breaks down protein is called pepsin. The inactive form of that protein is called pepsinogen, which is activated to pepsin through a proteolytic cleavage. And this HCl is there in the gastric mucosa to reduce the pH of the stomach, which is necessary because pepsin works better when the pH is between 2 to 3, okay? So there's an optimal pH for most enzymes, pH, temperature, depending on each enzyme has an optimum condition at which it catalyzes a reaction faster and better, So for this pepsin, it has been known that at pH 2 to 3, it's very, very active. It works very well. So the body has systematically accustomed itself to this. So it means playing around with this pH can compromise protein digestion in the stomach. Say you are sick, something that has to do with the stomach, and it has affected the pH of your stomach. This gastric mucosa secretes probably HCl that's not acidic enough for this pepsin to work, then it's going to be a problem, wouldn't it? Right? Looking at it from a research point of view. Okay. So in addition to acidity of gastric juice, probably denatures proteins in some uncooked foods. Okay. These are just some by the way explanation, extra knowledge for you. Okay. Pepsin rapidly attacks peptide bonds involving carbonyl group of phenylalanine, tyrosine, tryptophan. Now, before we start talking about this, I think it's worth showing. I should show you the picture of amino acid. You see, ammonia glutamate, okay? Ammonia, which enters the urea cycle and urea is excreted in the body. This is the reaction pathway that occurs for amino acid catabolism. First, there's a transamination reaction followed by deamination, removal of the amine group, right? And then the urea cycle. Okay, so these are your amino acids. All right, so you see, all the amino acids have the same basic structure. This is your carbon, carboxyl group, amino group. And then this is your R group, okay? This is serine. And you notice your threonine has a different R group, right? The R group here is different as per I. You see, the basic structure is usually the same, but there's a difference in the R group. Glutamine is here, okay? Special cases, cysteine, glycine, proline. These are also amino acids. Amino acids with hydrophobic side chains. These are examples. So you take your time, please. So you say phenylalanine, tyrosine, tryptophan. These are 3 amino acids that have aromatic R groups. This is an aromatic ring. This is a more of a benzene ring. We call it phenyl here, right? No? Benzyl. Phenyl. No, this should be a benzyl ring. This is a phenolic ring. And then this is an indole ring. Okay, is an indole ring. Now, somehow your pepsin works very well with proteins that have these amino acids in them: phenylalanine, tyrosine, and tryptophan. So that's what's being said here. Okay, the pancreas. The pancreas creates an alkaline mixture into the small intestine. Okay, so after the stomach, of course, the remaining undigested proteins or amino acids will go into your intestine, right? Where you have other proteases that also act on your, what do you call it, your protein. The mixture contains a number of proteases. There's trypsin in the intestine, chymotrypsin is also there. The trypsinogen, chymotrypsinogens, those are inactive forms that will be activated through some kind of proteolytic cleavage, okay? So trypsinogen is converted to active trypsin by enteropeptidase. So this enteropeptidase cleaves a particular bond in trypsinogen to convert it to active trypsin. Okay. The pancreatic enzymes to create oligopeptides and polypeptides, free amino acids, and short oligopeptides, which are transferred into the intestinal mucosa cells. Trypsin cleaves the peptide bonds involved in the carboxyl group of lysine and arginine. See, so we are seeing here trypsin, which acts on proteins in the intestine that prefers amino acids that with lysine and arginine. That's what we're saying here. You know, lysine and arginine, those are basic amino acids. They have a basic functional group. All right. Chymotrypsin is most active on peptide bonds involving phenylalanine, tyrosine, tryptophan. It has some similarities to pepsin, right? But the pepsin works on the carbonyl group, okay, while chymotrypsin works on carboxyl groups. So you spot the difference. Let's find out. If you have any questions, of course, you can post in the, what do you call it, the comments section, okay? So I think this is quite straightforward. That proteins are degraded in the cells by the free lysosomal proteins. The lysosome is like the sink where things go get degraded and prepared for excretion from the cell. Okay, so there are a lot of proteases or hydrolytic enzymes in the lysosome that takes care of this, and it also works for proteins that are to be read of are to be read from the cell. So example of essential amino acids, those amino acids you get mostly from exogenous sources: phenylalanine, tryptophan, histidine, valine, isoleucine, arginine, threonine, methionine, lysine, leucine. While those that can be secreted, synthesized by your body, the non-essential amino acids are here. Okay. All right. Okay. So tissue distribution of amino acid metabolism. The principal side of amino acid metabolism is the liver, of course. Okay. And then we talked about the breakdown of amino acid. Okay. This occurs in 3 steps: transamination, removal of amino groups on amino acids. It's accomplished by a variety of processes: transamination, oxidative and non-oxidative deamination, and followed by the urea cycle, indically. So what is this transamination? Transfer of amino group from an amino acid to an alpha keto acid or other amino acids. This is the amino acid, okay? This is your alpha ketoglutarate. An amino group was transferred from aspartate was converted to oxaloacetate, okay? Alpha ketoglutarate was converted to glutamate. The amino group present in aspartate was transferred to this glutamate. Basically, that's what happened here. Okay. So because if you look at this chart here, you see glutamate is what's converted to ammonia. So it means the body works better by optimizing operation, operates on the principle of maximum empowerment. Okay. So this reaction here is meant to transfer amino group from any amino acid to form glutamate, because alpha ketoglutarate can form glutamate. But for glutamate to be formed, you need an amino group from somewhere. And this amino acid has an amino group. So it forms the glutamate while it itself is being converted to oxaloacetate. Okay. So basically, this is what's happening here. And this reaction is catalyzed by aminotransferases that are dependent on a cofactor. Pyridoxal phosphate, that's vitamin B6, right? So you remember vitamins B and C are mostly coenzymes, right? Or cofactors, depending on how you want to use it. We mostly call it a coenzyme in this case. Okay, so this reaction, if I ask you to give me an example of a reaction that vitamin B6 plays a role as a coenzyme or cofactor, yeah, you can give me this reaction as an example, right? So the importance of transamination reaction is to collect amino groups from different amino acid in the form of L-glutamate. That's what I just finished saying, right? So L-glutamate is the target here, which will now be converted to ammonia going to urea cycle, which will be excreted through urine, okay? All right, you can just take your time. I think the basic explanation I have made already. Now, the second step, which is the deamination here, right, is the oxidative deamination reaction is catalyzed by glutamate dehydrogenase, okay? This is the major reaction in mammal cells through which ammonia is synthesized. So the reaction that converts glutamate now that has been synthesized from transamination to ammonia is catalyzed by glutamate dehydrogenase, right? That's the enzyme that does this. Okay. All right. Okay. So this statement here is simple, still linked to where alpha ketoglutarate acts as a sink for amino groups by accepting amino groups from various amino acids with the resultant formation of glutamate. Of course, to make sense, right? Alpha ketoglutarate is converted to glutamate here, but the amino group that formed the glutamate was gotten from this amino acid. So this serves as a sink to accept any kind of amino acid. What it needs from the amino acid is the amino group, basically. Get amino group. So this aspartate here can be any amino acid. Alpha ketoglutarate will collect the amino group and then convert the alpha ketoglutarate now to glutamate. So this statement is quite self-explanatory. So in mammals, all alpha amino acids can undergo transamination, except lysine and threonine. So these two amino acids cannot participate in this reaction. Okay, so you should find out why. The glutamate dehydrogenase in animal tissues occur in the inner matrix of the mitochondria and will utilize these coenzymes, but not both. Okay, NAD plus or NADP plus. Okay, all right, of course. The coupling of transamination and deamination reactions as nice by glutamic acid dehydrogenase account for most of the ammonia production in animals. So I think this is self-explanatory. Of course, you know how toxic ammonia is, so that's why it's immediately converted to urea, which will leave the cell. So these are things you can read yourself and understand. I've already explained everything that needs to be explained here, okay? All right. Ammonia intoxication include tremor, symptoms, slurred speech, blood vision, coma, and ultimately death. The body usually converts ammonia very quickly to urea to be excreted from the cell because ammonia is very dangerous. Okay. All right. Now, see if you remember again, some of these amino acids can be used as precursors for synthesis of sugar. I can dive into the TCA cycle. Okay, so it means some of these amino acids can be glucogenic. They can be used to synthesize glucose, or ketogenic, are used to synthesize glycogenic or glucogenic, that's used to synthesize glucose, or that can be ketogenic, that's used to synthesize fatty acids or ketone bodies. Okay, now the product of glycogenic amino acid can be pyruvate, oxaloacetate. Aspartate can be converted to oxaloacetate, right? Fumarate, succinyl-CoA, okay. Alpha ketoglutarate, proline, glutamine, glutamate, right? So try to make the connections, please, from previous classes and from what I even mentioned earlier. So, okay. Products of ketogenic amino acids, acetyl-CoA and acetoacetyl-CoA. Remember ketone bodies, right? Fatty acid oxidation. You should remember acetyl-CoA and acetoacetyl-CoA. Okay, examples of ketogenic and what do you call it? Glycogenic amino acid. These are examples of glycogenic amino acids that can be used as precursors for synthesis of glucose. Alanine, asparaginase, aspartate. I think that's a mistake. Asparaginase, aspartate, arginine, cysteine, glutamate, glutamine, stidine, methionine, proline, serine, threonine, tyrosine, tryptophan, valine. Phenylalanine. Okay. In ketogenic amino acids, there are two ketogenic amino acids, leucine and lysine. Okay. Then some amino acids can be ketogenic and glucogenic at the same time. An example of such amino acids are isoleucine, phenylalanine, tyrosine, and tryptophan. Ideally, we should go deep into how they do so, but I think for your own case, we're going to stop here. Just take your time and read. I think this is quite a very easy lecture. This is biochemistry simplified, to be honest. Okay. If you have any questions, you can please post in the comment section. Otherwise, I'll be expecting your assignment by tomorrow. And then hopefully tomorrow we're going to schedule a lecture again, this study session 2. All right. Study session 2 here is disorders of amino acid metabolism. It's a very, very, very short study session, so it shouldn't be a problem. All right, thank you, and see you in the next class.